So in the interest of time, I think let's get underway. I would like to thank you so very much for coming to our session today. My name is Michelle Bayou, and I'm the Research Grants Director at the Women and Children's Health Research Institute. I'm very pleased to serve as a session moderator for our presentation today. And on behalf of WICRI, thank you so very much for attending. I would like to start out the session in a good way by acknowledging that this conversation is taking place on Territory 6, Treaty 6 territory, the traditional lands of the Cree, Dene, Nakota Sioux, Saulteaux, and Blackfoot nations, and home of the Métis. I would also like to thank WICRI's funders, the Stollery Children's Hospital Foundation, and the Alberta Women's Health Foundation for making this research cluster possible. We would like to invite you to nominate your collaborators or networks to present the work they're doing in the research cluster. So if you would like to nominate anybody, please contact WICRI directly. We'll stick our email in the chat for you. And we will stay in the session at the end if you should have any questions about participating in the cluster. The session will be recorded, and we will post the link to the recording on our website in about 24 hours after the session ends. We're looking forward to sharing our research with you and encourage your questions after the presentation. So I would like to introduce to you Dr. Shokrollah Elahi. For those of you that know Dr. Elahi, he prefers Elahi as his name. So Elahi is a professor at the University of Alberta in the Faculty of Medicine and Dentistry in the Department of Dentistry. Elahi is a cellular and molecular immunologist who works on viral pathogenesis, immune regulation, cancer immunology, and immunotherapy. His research is focused on understanding mechanisms underlying immune dysregulation to generate protective therapeutic approaches. He obtained several degrees from the Tehran University of Medical Sciences in Iran and his PhD in immunology from Newcastle University in Australia. He also completed postdoctoral fellowships at the Vaccine and Infectious Disease Organization, or VIDO, and the Center for Infectious Diseases Research at the University of Washington in Seattle, where he explored HIV/AIDS pathogenesis. Further experiences at the Cincinnati Children's Hospital Medical Center led to his research interest in investigating the mechanisms underlying the susceptibility of neonates to infectious diseases. We'd like to invite Elahi to take the floor, and thank you so very much for your time today presenting.
Hey, thank you very much, Michelle, for the nice introduction. And I would like to thank you, WICRI, for this opportunity, and everybody who is attending this presentation. So I hope that throughout this presentation, I will provide convincing evidence that we have to pay attention to erythroid cells, not only as gas cargoes, but also because of their important immunological properties. So I don't need to explain about the role of RBCs. So the most important thing that we all know that RBCs are produced mainly in the bone marrow in adults through a complex interaction of different organs. However, if the bone marrow is unable to do its job, so this can happen in different circumstances. For example, in the context of chronic inflammation, cancer, severe anemia, hematological disorders, or if we have cancer metastasis to the bone, or for whatever reason that the bone marrow is unable to do its job, It means that it's unable to produce, for instance, RBCs. So body tries to find another location. So we will have extramedullary erythropoiesis that takes place in different locations, but mainly they are taking place in the liver and spleen. So it means that instead of bone marrow, liver and spleen are able to produce RBCs as a compensatory mechanism for the survival of the host. But the outcome of this process is the abundance of erythroid progenitors and precursors. Because normally when RBCs become kind of mature, they egress from the bone marrow. And but when we have extramedullary hematopoiesis, or even sometime when we have stress hematopoiesis, we have abundance of these erythroid progenitors and precursors in the periphery. So how we define these erythroid progenitors and precursors? So the most important thing I want to explain here is that just this definition of these cells. So RBCs, initially, like all other immune cells, they have nuclei. So erythroid progenitors still, they have their nuclei, and as they develop, they eventually become enucleated, and then they become full RBCs. So as you can see that they express two markers. One is CD71, another one is TER119 in mice, and CD235A in humans. So based on these two markers, we can define erythroid progenitors and precursors, CD71 and TER119 in mice, and CD71 and CD235A in humans. However, as you can see, there is another marker here, which is CD45, which is a leukocyte markers. It means that all immune cells express CD45. However, as you can see, as RBC become mature and they become, like full mature erythrocyte, they lose CD44, 45, and CD71, which is the transferrin receptor. So it's required for binding to transferrin and taking up iron. When these immature RBCs take up iron, then they don't need any more CD71. So CD71 gets downregulated. So on mature RBCs, we do not have CD71. We have only TER119 as erythroid lineage marker in mice, and CD235A as definite erythroid markers in human. That's why, based on these markers, we call erythroid progenitors and precursors CD71 positive erythroid cells, or simply we call them CECs. So throughout my talk, I just talk about CECs, and CECs means these are erythroid progenitors or precursors or CD71 positive erythroid cells. So about probably 15 years ago, I had no idea about these cells. And not only me, but also globally, nobody knew that erythroid progenitors have any immunological properties. So when I joined Cincinnati Children's Hospital, so I just proposed this idea that I talked to my supervisor that I think neonatal immune system is not what we have been thinking. And then so I started this project, which resulted in this publication in November 2013. And we basically discovered these cells. I mean, we named them as CD71 positive erythroid cells. And we showed for the very first time that these erythroid progenitors and precursors, they have immunosuppressive function. I briefly provide some kind of outline of what I did. One of the very first experiment that when I joined Cincinnati Children's Hospital, I did was just doing a flow cytometry staining on adult spleen versus a neonatal spleen. So I put all the immune cell lineages on one dye, one color, CD4, CD8 T cells, CD11B myeloid cells. These are dendritic cells, natural killer cells, and B cells. And I did a simple flow cytometry staining. And this was fascinating. In the adult spleen, I did see that about 95% of cells in the spleens are stained for these markers. That means that 95% of splenocytes in adult mice are different immune cells. But surprisingly, in POP, only 31% were immune cells. And then we had about 70% of cells, over 70, that we didn't know what they were at that time. So we had a huge proportion of cells that they were mainly present in newborns, and we didn't know what they were. So when you start a new project, you have no idea. It's not that easy. So I did lots of fishing experiment to figure out what these cells were. And finally, I discovered these are erythroid progenitors. As you can see, in adult, we have like a couple of percentage of them in the spleen, but they are massively abundant in the spleen of, this is a like 6-day-old, POP. So this was very interesting that these are abundant in newborns. So that was the beginning of our story. And then, okay, if you see something in mice, is it relevant to human? So we started looking at cord blood. And as you can see that if you look at adult blood, like us, if we are healthy, we don't have lots of this CEC in our blood. As you can see, less than 1% here in this case. And these are flow cytometry staining. So each dot represents a single cell. And as you can see here that in the cord blood, about more than 50% of the cord blood cells are these erythroid progenitors and precursors. And then we wanted to see that, okay, what happens during the development in neonatal mice? As you can see that it's interesting. We start looking at day 6, 9, and gradually these cells disappear in the spleen of neonatal mice. And the next question was, okay, what is the role of these cells? So we did extensive study. I just show an example here. So these are T cells stimulated. These are CD8, a subset of T cell stimulated. And then this is unstimulated cells. We measure the CFSC, which is a dye showing the proliferation of cells. So when cells are not stimulated, we don't expect much proliferation. When they are stimulated, we see like about 70% stimulation. When we add 1-to-1 ratio of these CECs to T cell, you can see that it comes down. And then if you add the ratio 2-to-1, it more significantly, as you can see in the cumulative data, there is a significant, dose-dependent immunosuppression effect on T cell proliferation. So we have done extensive study in terms of cytokine production, different assays. Overall, we showed that these erythroid progenitors or CECs have potent immunosuppressive properties. And I don't want to go through details. We have published extensively on these cells, but I just summarized. So now the question is that, okay, if these cells are immunosuppressive, how they do it? What's the mechanism of immunosuppression in these cells? So I can summarize that CECs utilize 3 different mechanisms to do their job. One is through cell-to-cell interaction. The CECs, they express molecules such as VISTA, such as PDL1, PDL2, and through interaction with their receptor, which is like PD1 and VISTA, they can suppress T cells. So one is through cell-to-cell interaction. We have published extensively. We have shown this mechanism. Another mechanism through soluble factors. We have shown that these CECs secretes arginase 1, arginase 2, TGF-beta, reactive oxygen species, and galactins. All these molecules or proteins are immunomodulatory. For example, arginase 1 and arginase 2, they deplete L-arginine in the environment, which is required for T cell proliferation. In the previous slide, I showed that they are suppressing T cells. So it means that they are depleting the microenvironment from what T cells need to proliferate with. So through soluble factor, they can suppress. And another interesting thing is that through this TGF-beta, which is a regulatory cytokine, they promote naive T cells become regulatory T cells. So regulatory T cells, from the name, these are immune regulator. They are policing the immune system. And in addition, these erythroid progenitors, not only they have immunosuppressive function themselves, but also they promote another subset of regulatory T cells in our body. So this slide demonstrates that the CECs utilize different mechanisms to mediate their immunosuppressive function. This was all in vitro. In the lab, we have shown that they are using this mechanism. How about in vivo? If we go to the inside the animal, what we can learn. So one of the things that initially I did was just depleting these cells, because these are erythroid progenitors. We cannot 100% deplete them because it would be fatal. These are RBC progenitors. But we can use anti-CD71, which is the transferrin receptor I mentioned. If you use antibody against it, you can do partial depletion. You can see about 50% reduction. And then we wanted to see that, okay, if we even partially deplete these CECs, what happens when we infect neonatal mice? So this is the number of bacterial colony forming units in the liver and in the spleen of pups infected with Listeria monocytogen, because it's an important pathogen that infects newborns. So as you can see that when we even deplete these CECs, there is a significant reduction in the proportion of, these bacterial colonies. It shows that these cells in vivo have immunosuppressive function, and their abundance is associated with susceptibility to infection, because when we deplete them, they become more resistant to infection. We have done extensive studies. This is just an example. We have done E. coli infection. These are systemic infection. We have done even whooping cough infection, respiratory infection, and we have shown that these cells have immunosuppressive and make newborn more susceptible to infection. So these observations suggest that these cells are abundant in newborns and provide an immunosuppressive microenvironment in newborn. So to prove that, one of the things that we did was doing adaptive transfer. So we transferred total splenocyte from adult mice into pups. And then, as you can see, that we have just adult cells. We have adult cells that are adaptively transferred into the pups, and these are just pup cells. So there is no infection, and we look at a cytokine which is very important against Listeria monocytogen, which is tumor necrosing factor alpha, TNF alpha, and then following infection, we look at the cytokine production, TNF alpha, by these cells. These are myeloid cells like CD11B. These are neutrophils and monocyte macrophage kind of population. So, and as you can see, when there is no infection, there is a background, little amount of TNF alpha production. However, when we infect adults, you can see that adult cells within the adult microenvironment, they produce like 15% this cytokine. However, when we put the adult cells in the neonate, they act like neonatal cells. This production, as you can see here, cumulative data, so there is a significant difference when we put adult cells within the neonatal microenvironment. However, when we have infection, as expected, because neonatal immune system is suppressed, so we don't see TNF alpha production when you compare no infection versus infection, because their immune system is highly suppressed. But the most striking was that adult cells within the adult environment function very well, but when we put them in the neonate, they function like neonatal cells. It means that neonatal environment is immunosuppressive that prevents adult cells from doing their function. So this observation overall showed that neonatal immune system is a more suppressed microenvironment. And this is a fact that we all know that newborns are more susceptible to infection. Unfortunately, there is lots of mortalities under age 5 globally still, based on even recent WHO report. So, and in the past, we have had this kind of concept because neonates are deficient in their, lots of their functions. So we have been thinking that neonatal immune system is like their body is underdeveloped or immature. However, this study kind of challenged the old dogma, and we challenged the neonatal immune system immaturity, and we provided a new concept that neonatal immune system is highly suppressed. And we have done the other way around. So if you put the neonatal immune cells, like isolate those CD11B, put them in adult, and infect the adult, they function like adult cells. So this demonstrates the neonatal immune system is highly suppressed, which is not consistent with the old dogma that they are immature. They are not immature, but there is an active immune suppression in place. And this has been something that recent years, there has been received lots of attention. Other scientists, they have come to the same conclusion as us, that neonatal immune system is not immature, and immaturity is kind of unacceptable. But now the major question is that, okay, why neonatal immune system needs to be suppressed? So it doesn't make sense that God or the creator created these newborns with a suppressed immune system that they become miserable. They become all the time sick, and there might be a greater benefit into it. So we try to understand what is the greater benefit. So we did extensive studies, but one of the things that we look at was looking at the gut. So what happens in the gut of these newborns when we deplete these CECs? So this is just similar to the plot I showed before, but this is a cumulative data. These are control pups. These are anti-CD71 treated. So we see significant depletion of these CECs when we treat pups. And then when you look at the gut, these are CD11B, which is, as I said, myeloid cells, and CD11C, these are dendritic cells. And we see that if we deplete them at day 3 and a couple of days later, look at the TNF alpha, which is an inflammatory cytokine in the gut, we see that there is, compared to the control, you see that average of 5-fold increased TNF alpha. So elevation of TNF alpha is associated with inflammatory response in the gut. And this, under normal circumstances, we should not have such an inflammatory response to the microbial community in the gut. So, but when you do this in germ-free mice, you don't see this. In germ-free mice, there is no inflammatory response if you deplete these CECs. So this suggested that there is a crosstalk between germs in the gut and CECs. So it means that CECs are suppressing the immune response in the gut. So I came up with this kind of cartoon. So I published in Trends Immunology. I proposed this idea that CECs from the spleen home to the gut. And under normal circumstances, we have lots of CECs, these red cells, are in the gut because they have immunosuppressive function. So they provide a suppressed microenvironment, and everything is intact, and we have commensal bacteria in the gut. But if we deplete these CECs are gone. And then immune system becomes out of control, and they become activated in response to microbial, byproducts. And then we have inflammation, as I showed, by TNF alpha, and this inflammation results in, eventually dysbiosis and gut leakiness. So this was a proposal. We didn't prove it, but just I proposed. Until we started addressing this idea, is this the case or not? This was the work that mainly by Don, other people helped, but led by Petia, my former lab member. And so one of the things that I asked her to do is just what we did during the development to see that the trend of these CECs. So as you can see that CECs, I showed you from day 6 to adulthood, but then we were curious to go further back. If we start from day 1, look at the pops to see what is the trend of this CEC. It was interesting. You can see that day 1 start going up, they max day 6, 9, and they decline. And then we start looking at the gut. It was very interesting. If you look at the small intestine, they are max at day 1, and then they gradually decline. But if you look at the colon, there are very little, and we don't see much changes except around day 21, 23 that they get weaned, and there is a diet change probably influences the microbiome, and then we see a kind of significant increase in CECs. But the striking observation was here, that the CECs in the small intestine are max at day 1, but in the spleen, they are max at day 6 and 9. So we thought that, as I showed in the previous slide, I suggested that CEC home to the gut. But this might not be the case because the frequency pattern is different. And then we did extensive studies phenotyping them, looking at different markers on CECs from the gut versus a spleen, and they were completely different. So these observations suggested 2 possibilities. One, that these cells come from the spleen to the gut, and they gain gut-associated phenotype. They had like gut homing receptors, and they were more potent immunosuppressive. The other possibility was that maybe they are generated in the gut. They are not coming from the spleen to the gut. So, and we did extensive study to make it kind of short. We found that in the gut, we have central macrophages. So central macrophages are CD11b negative, CD169 positive, F480 positive. So this population here. This is the bone marrow. We expect to have central macrophages because central macrophages like a nursing mom for immature RBCs. And you can see that they are highly abundant in the gut of newborn. And they also express this marker, which is VCAM1, close to 100% of them. So these observations suggested that erythroid cells are maybe produced in the gut, where they need to be to provide an immunosuppressive microenvironment to the microbial communities. To confirm this, we did image stream. So this is a combination of flow cytometry and kind of microscopical examination. So you can see that in the center, we have the central macrophages that are expressing CD169 and F480. And these are erythroid cells, or those CECs that express because we had limitation in terms of number of antibodies we can use. So, and if you look at them, you can see that this kind of here, that in the middle, we have this central macrophages, and is surrounded by those, erythroid cells. And then when we submitted the paper, reviewers were not convinced. They wanted to do immunofluorescence on the tissue. And this is more beautiful. And so you can see that this is central macrophages in the middle. And then these CECs are surrounding it. So the role of these central macrophages, as I said, is like a nursing mom. So there is a tight interaction between these. And these central macrophages, they recycle iron from dying old RBCs, and they giving it to these maturing CECs. And so this was for the very first time we demonstrated in the neonatal gut, we have erythropoiesis niches. So we have shown, in the paper exactly where they are located. So this was very fascinating that erythropoiesis takes place in the gut. So now the question is that, okay, now we have erythropoiesis in the gut, we have CECs in the gut. So now if we deplete the CECs, what happens to the microbial communities? As I said, when we deplete CECs in the germ-free, we don't see any inflammatory response, but in wild-type mice, we see. So this was a very fascinating observation. So when we deplete CECs, you can see that there is a massive changes in the microbiome. This is a PCA plot. The blue are just control mice, and these are anti-CD71 treated. So when we deplete CECs, these are clustering together, and these are clustering together. These are 3-day-old pups, and then 2 days after, we collected the fecal sample for 16S ribosomal RNA, and you can see that these are individual pups, and this is cumulative data. They are completely different. So depletion of CECs result in dysbiosis. So if you want to go in further analysis, you can see that when we deplete CECs, the number of bacteria are increased in the gut, and they are more diverse. And some of them, you can see that Staphylococcus goes up, Lactobacillus significantly goes up, and Enterobacteriaceae comes down. So it just shows that there is dysbiosis. So this demonstrated that CECs are involved in symbiosis. When they are depleted, we have changes in the diversity and number of bacteria in the gut of neonates. So now the question is that, because we did it 2 days after depletion, is it transient or this lasts for a long period of time? So to address this, so this is something that we have to do more studies in the future, but one of the experiments we did the same depleted CECs at day 3, and 32 days later, so we kept pups in the same cage. We removed mothers because the cage effects can influence. So they were just mating, all they were in one single cage. So as you can see that, we look at their sexes. So control female, control male, CEC depleted female, CEC depleted male. So first of all, you can see that males have more bacteria in their gut, and they are more diverse. This is other people already shown that this is the case. Males have more bacteria and they are more diverse compared with females. But the interesting part that we observed was that when we deplete CECs at day 3, 32 days later, we don't see any difference in males. There is no significant in terms of number of bacteria in males, in terms of diversity. However, we see a significant difference in the number of bacteria and the diversity in females. So this was very interesting. It shows a sex-dependent effect. When we deplete CEC and look at them, like over a month later, there are differences mainly in females in terms of diversity and in terms of the number of bacteria. But if you go in more details, you can see that the number, you can see based on the control, obviously, you can see that the red area is significantly, reduce anti-CD71, you can see here, but Some of other bacteria, they did not reach complete, significance. They are very close, 0.055, but they are not significant. But it shows that there are changes. So because we have a small number of animals, but in males only, we did see that firmicutes becomes significantly reduced when we deplete CECs. So this overall shows that when we deplete CECs in the neonatal period, even there are potentially long-term effects, which is more pronounced in females. So then when we submitted, the reviewers asked why they are more pronounced in females. So we did lots of investigation and we found this is the reason. Females have more CECs in their gut than males. So these are neonatal, day 3 and day 6. As you can see that female have more. So in summary of this section, we have shown that CECs are highly abundant in the gut and the spleen of neonatal mice. They have immunosuppressive properties and they allow swift adaptation of the microbiome, because if we deplete them, results in dysbiosis. So this was all great. So how about human newborns? It was very difficult, almost impossible to recruit neonatal cohort here, and I tried with the help of Lori West for several years. We could collect only 3 samples. So then I approached somebody in Mexico, and then we did the study in human newborns in Mexico. And that was the case. Still, we see that these CECs are highly abundant in newborns. You can see day 2, day 8, day 60, and this is the cumulative data that they are significantly higher. They are coming down, but they are still significantly higher throughout the development compared with 6 to 12 months and 1 to 5 years. So this shows that not only we see CECs are abundant in mice, but also they are abundant in humans. And actually, I recently saw another paper that confirmed this in a Korean cohort, that CECs are abundant in human neonates. So now this is all, it's interesting, but obviously we cannot do any, intervention studies in newborns. But then we went to cord blood to see that if there is a difference between full term versus preterm. So this was the thing that done in Cincinnati Children's Hospital. Actually, I did it when I was there. But if you look at the gestation weeks, you can see that we look at whatever we had access to from 26 weeks to 42, especially in 26, 29, 31, and 32 weeks of gestation, there is a significantly lower number of CECs and compared to full gestation. So this suggests that maybe preterm newborns, due to lower CEC, might be predisposed to dysbiosis and necrotizing enterocolitis. This is just a hypothesis. This is just a suggestion. We need to address this. But so the presence of these cells in the cord blood suggests that maybe they are also present in placenta. So we have started doing another study to see that what is the role of these cells during pregnancy. So these are human samples. So human placenta tissue, you can see that they are abundant in placenta and they are abundant in the cord blood. And non-pregnant mothers don't have much, but during pregnancy, these CECs are expanded in mothers, as you can see this. And then we wanted to do some functional assay to understand their role, so we switched to animal model. And as you can see here, this is a non-pregnant mouse looking at the spleen. And during the pregnancy, these CECs are expanded in the spleen. And also, similar to human, placenta is enriched with CECs. And even we can see some of these cells in the uterus. So then we wanted to see what is the role of these CECs during pregnancy. So we took the same approach. We tried to deplete them. So with anti-CD71, and then we depleted them. I think I had a slide, no. So around 12.5 and 13.5 gestation days. So we depleted them. So if you inject anti-CD71, we see that these are gone in the spleen and even in the placenta. So there is a significant reduction of CECs by treating them with anti-CD71, and the outcome was complete resorption. So these are control, and this is each pop, and when we deplete them, there is either complete resorption or partial resorption. You can see that there is only some tissues left and with the placenta. So this study, I mean, I don't go in the mechanistic part of it because it's published several years ago, but we showed that basically CECs are important in fetal-maternal tolerance. And then we switched to human, another study that we did with the help of Vivian, which unfortunately moved to Toronto, but my graduate student, Garrett, led this study. So we did collect blood from preconception, from mothers throughout the pregnancy and postpartum. So this was interesting that during pregnancy, you can see that in the third trimester, there is a significant increase of CECs, and they max at third trimester. So second, there is significant with the preconception. But then we wanted to look at IBD to see what happens in IBD mother versus healthy mother. And it was very interesting. In the IBD mother, we don't see this expansion that normally we see in healthy. So there is a significant reduction of these cells in the blood circulation of mothers during pregnancy in Crohn's disease or ulcerative colitis versus healthy mother. So this might explain the problem that we see during pregnancy, because lots of autoimmune diseases like MS, like arthritis, when mothers become pregnant, they benefit from pregnancy because their immune system becomes more regulatory, anti-inflammatory, but this is not the case in IBD. Might be one mechanism that these regulatory cells are not expanded. And then we wanted to switch to mice to understand that what happens if during the pregnancy, these CECs are not expanded. So we use the same approach, depleting them, and then looking at the interleukin-6, inflammatory cytokine, and TNF-alpha. As you can see that if during the pregnancy we deplete these CECs, we have massive production of these inflammatory cytokines in both CD11B and CD11C. And then it becomes, the gut becomes leaky. So following treatment, we can feed the mice with fitsidextran, and 2 to 4 hours after, we can collect the blood and measure the translocation of fitsidextran to the plasma, so we can show that if there is a gut leakiness. So this observation showed that a lack of expansion of CEC potentially in IBD patient, is associated with more inflammatory response and the leakiness in the gut. So if I want to summarize the second section of our observation, we found the CECs are present in the blood circulation of human newborns. They are present in the cord blood, but preterm baby have lower abundance of these cells, and they become expanded in the placenta, cord blood during pregnancy, and they are very important in maintaining fetal-maternal tolerance. If we deplete them, it will end up an inflammatory response and gut leakiness in pregnant mice. And other people have shown the same observation that preterms have lower, CECs in their cord blood. And we had an observation. This is a single observation. It's very hard to tell, but it was a case record. We had a case report that we had twins that one of them was delivered naturally, the other one by C-section. And the one that was 21 minutes after was born by C-section has significantly lower CECs in its placenta and cord blood. So this is just a case report, but it was very interesting. So I talk about the depletion of CEC, which is impacts on the gut leakiness. So now we wanted to see that, okay, if it depletes CECs, whether it modulates the microbial community during pregnancy in mice. So this is just a normal mice. Adult mice, we don't see much of CEC in different components except in cecum and colon. We have more CEC, we see more CECs in this compartment. So if mice become pregnant, interestingly, you can see that there is a significant increase in CECs in the cecum and colon. So this suggested that maybe they do the same role as newborn. Maybe they play an important role in symbiosis in the gut. So we tried to do a similar experiment. So we did, check for a vaginal plug to kind of have a pregnancy timing, and then we treated them at E12.5, 13.5. We did either one injection and euthanized mice 1 day after or 3 days after treatment. And we collected the fecal sample for sequencing. And we found that, this is other people have shown that non-pregnant mice have different microbiomes or microbial communities compared with the pregnant, untreated. But the interesting was that when we treat mice with anti-CD71, that means we deplete CECs either 1 day after treatment or 3 days, they cluster together. So we took them all together in terms of analysis, and this is what we found. It was very interesting that when we deplete, obviously, as I said, pregnant control with non-pregnants are different. This is anticipated. But the most important part was when we deplete CECs, there is dysbiosis. The diversity and number of bacteria are changed in, pregnant mice compared with just the normal pregnant versus CEC depleted. So this suggests that CECs also play an important role in symbiosis during pregnancy. If we deplete them, they result in dysbiosis. So if you have dysbiosis in the gut, you can end up having inflammatory response. So we look at some of the genes associated with inflammation, for example, interleukin-6, this is a chemokine, CXCL1. These are toll-like receptors that they can respond to the microbial communities, and TGF-beta, which is a regulatory cytokine. As you can see, the red one is anti-CD71 treated. So when we deplete CEC, there is more signs of inflammatory response in the gut and lower TGF-beta, which is a regulatory anti-inflammatory. So this shows that how important these CECs are during pregnancy. So even beyond pregnancy, if you look at even mice, male and females are different. Females significantly have more either in biopsy or black cysts than males. So this is very important. If you have more regulatory T cells, cells in females, and then you want to do any studies and you decide using male versus females, this can have significant impact on the outcome. This is also the case in humans. If you look at male versus female, so female in general have more CECs. And we also, because this was during the COVID, we had very few Ns to look at pre and post menstrual blood from the same individual, but it shows a trend that there is a significantly increase in CECs. So that might explain some of the differences that people feel. They have become more probably susceptible to infection because they have more of this immunosuppressive. And this, not only the numbers, but also the functionality of these CECs are different in male versus female. In female, we have more CD45 positive. If you remember on my third slide, I showed that CD45 is more on erythroid progenitors. So erythroid progenitors have more potent immunosuppressive. If females have more of this 45, that means that these cells have more immunosuppressive function. I mean, we don't see the frequency, but the absolute numbers are higher in both black 6 and biopsy. And as you can see, this is just an example of reactive oxygen species and arginase 1. 45 positive have more, and females have more 45 positive. It means that these cells not only are more in females, but they have more potent immunosuppressive function. So we cannot talk these days about anything in science without mentioning long COVID. So I end up with long COVID. So you have I probably experienced, definitely experienced long COVID myself, but some of you guys have heard. But so we have published this last year, but one of the things that we observed in our cohort that 75% of long COVIDs are females. And one of the things that we found in long COVID, we see expansion of these CECs in long COVID patients. And one of the novel things that we discovered that these CECs, I didn't talk about it in newborn. That's something that I'm going to submit a grant to Vickery soon. So, but for long COVID, we see that these CECs secrete artemin. Artemin is a neurotrophic factor. And this is incredible that erythroid progenitors secrete this neurotrophic factor. So this neurotrophic factor is very important in pain based on the literature and also tumor progression, especially in breast cancer. But in the neonatal period, the only cells that express artemin receptors are microglia. So we believe that this erythroid progenitor, by secreting artemin, may influence brain development in newborns, but in the pathological condition, the story is different. So when we look at the plasma of long COVID patients, we see massive elevation of artemin in long COVID patients, as you can see here, compared with those that got COVID and recovered without any symptoms. So, but another observation that we did correlation of studies between the artemin levels with two pain indexes. One, severity of the pain, and then wide pain spread index. And as you can see that there is a strong correlation between artemin level and pain indexes in long COVID. And more importantly, there is a very kind of more than moderate, 67% of cases can explain that there is a positive correlation between artemin and cognitive impairment or brain fog. So this illustrates that how these erythroid progenitors are important beyond the neonatal, beyond the pregnancy period. And this is an angle that I just wanted to share with you guys. So I hope that throughout this presentation, I tried to kind of provide some insight that these cells have been neglected. They have, I mean, very potent immunoregulatory properties, and they are very important in the adaptation of microbial communities in newborn and pregnancy. And we should pay attention in different physiopathological conditions. My lab, for example, is working on these cells in the tumor because we see that in the context of tumor also these cells are expanded, they are in the tumor microenvironment, and so we are trying to modulate them. So As you can see that these cells have been neglected, but we have been working on them, and I hope that I was able to convince you guys that these cells are important and we should take them into consideration in any model that we have. So I would like to acknowledge lots of my former current lab members and collaborators that they have been vital to do these studies. And most of the work that we have done during pregnancy, newborn have been funded by, Vickery, and I had one CIHR grant on this study, most of them from Vickery. I thank Vickery for continued support, and thank you very much, everyone. And with that, I'm happy to answer any potential questions.
Thank you so much, Elahi. It was absolutely enlightening. And honestly, had about 3 questions, but you answered everything. Sandy, do you have a question?
I do. I'm sitting here at a conference. I hope you can hear me.
Yeah.
Okay, good. Even though I look like I'm at the U of A, I'm actually in North Carolina. That was great. Thank you. Really fascinating. Some of my questions were answered as well, but I'm really intrigued about the male and female. So I'm going to work backwards just because long COVID was the last thing you said. You talked about the females, and more females have long COVID and Artin going up, but what about the males? Did you have any males in that long COVID study? And
Yeah, we have so we have done lots of work. Obviously, we have a paper, Cell Reports Medicine, which is under review. We have looked at the role of sex in this concept. But yes, we had males, we had females. We see also elevation in males, but artemin can be sourced from endothelial cells as well.
Huh.
So that means that the obviously neuronal cells as well. So neuronal cells, endothelial cells, but in terms of immune cells, we have done all the immune cells one by one in human and mice. None of them even have the gene for artemins. So the only cells that they have gene for artemins are these erythroid progenitors. And another thing that we have tried in different contexts, but I didn't explain it, but mice CECs, the membrane of mice CECs is different. We can do intracellular staining on them, but human CECs, we cannot because they get lysed. So because these long COVID patients and even acute COVID patients, we have seen that the membrane of these CECs become rigid. That's why we have been able to do intracellular staining on this. So we have mentioned that this is kind of happening. Actually, there was another group published in Blood a couple of years ago. They showed that the effect of SARS-CoV on the membrane. So that's the only groups that we have been able to look at inside the CECs are acute COVID and long COVID. But yes, males also have elevated, but it's significantly lower than females.
Because they start off lower? With the CECs are lower
Yeah, they have lower CECs.
So we start lower.
Yeah, they have absolutely lower CECs compared to female, but even it might be sourced from other places, but overall they have significantly lower than female.
And then my other question, if I can have one more question. Okay, it's looking at Michelle. Okay. But it goes back to and I forget even exactly what it was, but you were looking at newborns in Mexico, right? Because you were trying to get them here, but you didn't separate them by sex. I forgot was that
That's a very good question. Actually, I mean they missed that, and then they said that we could not go back ethically to figure out if they were male and female. So they collected samples, they did the analysis, but then when I asked them the sex, said that we cannot ethically go back to the file, and we missed that.
That would have been fascinating.
Yeah.
And then another question, I'm just thinking about it now, is you were saying with the increased CECs, it's immunosuppression, and that allows for more cancer. Now I'm going to ask a cancer question. So specific cancers and cancers that women are more susceptible or have a greater incidence proportionality because
I
of
think
CECs? Just trying to.
we have published one paper, I think was last year. We did melanoma, but it's more, especially when you look at the role of artemin, is more breast cancer. So because I mean, actually, I have 2 graduate students working on this project, and I have a CIHR grant on this. So what we see, or I mean, actually, we have generated also artemin knockout mice. So we had artemin knockout mice. That's a great tool that we want to address in the context of newborn and also cancer. So it appears that because tumor cells have artemin receptor in the context of breast cancer. So, but they have, especially triple negative breast cancer, they have artemin receptor. So when there is artemin available, the interaction of artemin with its receptor results in tumor progression and metastasis.
Wow. Fascinating.
Yeah, it's just crazy. I
Wow.
mean, you don't expect, I mean, erythroid progenitors can be so problematic. Yeah.
Exactly. Well, cool. Well, thank
I mean,
you.
it can be beneficial depending on the context, but yeah.
Right, exactly. Yeah. Well, it's always that, right? So thank you. Really
Oh,
appreciate the
no,
time you
thank you
took
very
to
much.
share all this with us.
Thank you for great questions.
Oh, Joseph Ting. Oh. There he goes. I'm not sure if you can see the chat, but Michelle can read it out.
So it says fascinating talk, great insights on the sex differences in outcomes of preterm infants. Thanks very much. So of course we will be posting the recording to our website, and we'll encourage the cluster to refer to the website for the recording. If we get any questions for you from the recording that is posted, Elahi, we will forward them to you for your consideration.
Well, thank you very much for the opportunity and
thank you so much for
for
your
all the
time.
support. Okay, thank you very
Thank
much.
you. Have a great day.
You too.
Bye, everyone.
Bye.